Proximate analysis is a broader analysis of food because when we talk about the food matrix, it includes elements like carbohydrates, fats, proteins, moisture, etc. And when these groups are joined together in a broader sense, the analysis is called as proximate analysis. It is called proximate because it doesn't measure the very exact amount of these elements together like carbohydrates or fiber or moisture. So it might have closely related group rather than exact amount of one component of food. So that is why it is called as proximate analysis. Like crude fiber is one element of proximate analysis. It refers to the organic residue of vegetable origin. It consists of elements like cellulose, lignins, and pentogens. As I said, that food is complex matrix, so we have to actually remove other components like carbohydrates and fats by treating them with alkalis or acids, and then whatever is left is termed as crude fiber. And that is the reason we use the term crude, because it is not the exact amount of fiber. It might have some other elements present in this analysis as well. Moisture content is another important element of proximate analysis. And every food would have a particular amount of moisture present, like fruits and vegetables might have a higher amount of moisture as compared to pulses or dried milk where the moisture content is not very high. It measures the stability and quality of food. So this term, moisture content, is very often used for a variety of reasons in food analysis. Because there are different kinds of foods and they have all different kind of structures as well as the different forms of water is present. So we have to have very different methods for every type of food for the removal of this moisture. The next term is called crude fat. Again, it is called crude because it's not exact fat. It is generally also called as ether extract. So whatever is dissolved in ether from the food matrix is called as crude fat. It includes fat contents. It can also have other things like phospholipids, lecithin, sterols, fatty acids, waxes, etc. So that is again the reason why it is called crude. Ash content is another important element of proximate analysis where we measure the mineral contents present in the food. And it is normally achieved by burning all the food, and whatever is left that cannot be further burned down, it is called as ash. And in the case of ash, these minerals might change their chemistry into oxides or carbonates, phosphates, etc. And some of these new compounds, they might be volatile. That would mean they would be evaporated and we cannot measure them gravimetrically. So that is why we should take extra care to follow the instructions for every different kind of food for their ash analysis. Another term is called crude protein. Again, we use the term crude because it is not the exact protein. We do measure nitrogen content in food, and that nitrogen content is very much related with protein contents, but there could be other compounds which have got nitrogen in them, like some of the lipid nitrogen, nucleic acids, and vitamins, etc. So when we measure the nitrogen content, we are measuring the nitrogen content present in proteins as well as in other compounds which are found in food. And this nitrogen is again multiplied by a specific factor for every kind of food to get the protein content. So we are not actually measuring the protein directly, rather it's an indirect way of measuring protein content by measuring the nitrogen. And this nitrogen has got different sources. So the term is again crude protein.